Hi class, this is your instructor, Skylar Huff, and we're now in chapter 14. This is evolution and natural selection. This is the last chapter of the course. Let us begin. The great diversity of life on Earth, ranging from bacteria to elephants and roses, is a result of a long process of evolution. It is the change that occurs in organisms' characteristics through time. In 1859, the English naturalist Charles Darwin first suggested an explanation for why evolution occurs, a process called natural selection. Biologists soon became convinced Darwin was right and now consider evolution one of the central concepts of the science of biology. In this chapter, we will examine Darwin and evolution in detail, as the concepts we encounter will provide a solid foundation for your exploration of the living world. The theory of evolution proposes that a population can change over time, sometimes forming a new species. And a species is defined as a group of populations that possess similar characteristics and whose members can interbreed and produce fertile offspring. This famous theory provides a good example of how a scientist develops a hypothesis, in this case, a hypothesis of how evolution occurs, and how, after much testing, the hypothesis is eventually accepted as a theory. Charles Robert Darwin was an English naturalist who, after years of study and observations, wrote in 1859 one of the most famous and influential books of all time. This book, On the Origin of Species by Means of Natural Selection, created a sensation when it was published, and the ideas of Darwin expressed in it have played a central role in the development of human thought ever since. In Darwin's time, most people believed the various kinds of organisms and the individual structures resulted from actions of the Creator. Species were thought to be specifically created and unchangeable over the course of time. In contrast to these views, a number of philosophers have presented the view that living things must have changed during the history of life on Earth. Darwin proposed a concept he called natural selection as a coherent, logical explanation for this process. Darwin's book, as the title indicates, presented a conclusion that differed sharply from the conventional wisdom. Although his theory did not challenge the existence of a divine creator, Darwin argued that this creator did not simply create and then leave them forever unchanged. Instead, Darwin's God expressed himself through operation of natural laws that produced change over time. And that is evolution. The story of Darwin's theory begins in 1831 when he was 22 years old. The small British naval vessel, the HMS Beagle, was about to set sail on a 5-year navigational mapping expedition around the coasts of South America. The red arrows shown in figure 14.3 indicate the route taken by the HMS Beagle. The young 26-year-old captain of the HMS Beagle, unable by British naval tradition to have social contact with his crew, and anticipating a voyage that would last many years, wanted a gentleman companion, someone to talk to. Indeed, the Beagle's previous skipper had broken down and shot himself to death after 3 solitary years away from home. On the recommendation of John Stevens Henslow, one of his professors at Cambridge University, Darwin, the son of a wealthy doctor and very much a gentleman, was selected to serve as the captain's companion, primarily to share his table at mealtime during every shipboard dinner of the long voyage. Darwin paid his own expenses and even brought along a manservant. Darwin took on the role of the ship's naturalist. The official naturalist, a man named Robert McCormick left the ship before the first year was out. During this long voyage, Darwin had the chance to study a wide variety of plants and animals on continents and islands in distant seas. He was able to explore the biological richness of the tropical forest, examine extraordinary fossils at the southern tip of South America, and observe the remarkable series of related forms of life on the Galapagos Islands. When Darwin returned from the voyage, at the age of 27, he began a long period of study and contemplation. During the next 10 years, he published important books on several subjects, including the formation of oceanic islands from coral reefs and the geology of South America. He then devoted 8 years of detailed study of barnacles, a small group of shell marine animals that inhabit rocks and pilings, eventually writing a 4-volume work on their classification and natural history. In 1842, Darwin and his family moved out of London to a country home at Down in the county of Kent. In these pleasant surroundings, Darwin lived, studied, and wrote for the next 40 years. One of the obstacles that had blocked the acceptance of any theory of evolution in Darwin's day was the incorrect notion, widely believed at that time, that the Earth was only a few thousand years old. The discovery of thick layers of rocks, evidence of extensive, prolonged erosion, and the increasing numbers of diverse and unfamiliar fossils discovered during Darwin's time made this assertion seem less and less likely. The great geologist Charles Lyell, whose Principles of Geology Darwin read eagerly as he sailed on the HMS Beagle, outlined for the first time in the story of the ancient world of plants and animals in flux, species constantly becoming extinct while others were still emerging. When the HMS Beagle set sail, Darwin was fully convinced that species were immutable, meaning that they were not subject to being changed. Indeed, it was not until 2 or 3 years after his return that he began to seriously consider the possibility that they could change. Nevertheless, during his 5 years on the ship, Darwin observed a number of phenomena that were of central importance to him in reaching his ultimate conclusion. In the rich fossil beds of South America, he observed fossils of the extinct armadillo that you'll see in figure 14.4. They were surprisingly similar to the armadillos that still live in the same area, shown on the left. Why would similar living and fossil organisms be in the same area unless the earlier form had given rise to the other? Later, Darwin's observations would be strengthened by the discovery of other fossils that show intermediate characteristics pointing to successive change. Repeatedly, Darwin saw the characteristics of similar species varied somewhat from place to place. These geographical patterns suggested to him that organismal lineages change gradually as individuals move into new habitats. On the Galapagos Islands, which is 900 kilometers off the coast of Ecuador, Darwin encountered a variety of different finches on the islands. The 14 species, although related, differed slightly in appearance. Darwin felt it was most reasonable to assume that all these birds had descended from a common ancestor blown by winds from the South American mainland several million years ago. Eating different foods on different islands, the species had changed in different ways, most notably in the size of their beaks. The larger beak of the ground finch in the upper left of figure 14.5 is better suited to crack open large seeds it eats. As the generations descended from the common ancestor, these ground finches changed and adapted, what Darwin referred to as descent with modification. In a more general sense, Darwin was struck by the fact that the plants and animals on these relatively young volcanic islands resembled those on a nearby coast of South America. If each one of these plants and animals had been created independently and simply placed on the Galapagos Islands, why didn't they resemble the plants and animals of islands with similar climates, such as those off the coast of Africa? Why did they resemble those of the adjacent South American coast instead? It is one thing to observe the results of evolution, but quite another to understand how it happens. Darwin's great achievement lies in his formation of the hypothesis that evolution occurs because of natural selection. Of key importance to the development of Darwin's insight was his study of Thomas Malthus's essay on the principle of populations. In his book, Malthus pointed out that populations of plants and animals, including human beings, tend to increase geometrically, while their food supply increases only arithmetically. In a geometric progression, the elements increase by a constant factor. So if you look at figure 14.6, it shows the progression from 2 to 6 to 18 to 54, where each number is 3 times the preceding one. In arithmetic progression, in contrast, the elements increase by a constant difference. That red line shows that. The progression proceeds as 2, 4, 6, 8, where each number is greater than the preceding one, and where each number is 2 greater than the preceding one. Because populations increase geometrically, virtually any kind of animal or plant would cover the entire surface of the world within a surprisingly short time if it could reproduce unchecked. Instead, population sizes of species remain fairly constant year after year because death limits population numbers. Malthus's conclusion provided a key ingredient for Darwin to develop the hypothesis that evolution occurs by natural selection. Sparked by Malthus's ideas, Darwin saw that although every organism has the potential to produce more offspring that can survive, only a limited number actually do survive and produce further offspring. Many examples appear in nature. Sea turtles, for instance, will return to the beaches where they hatched to lay eggs. Each female will lay about 100 eggs. The beach could have been covered with thousands of hatchlings, such as you see in figure 14.7 in your textbook on page 255, trying to make it to the water's edge. Less than 10% will actually reach adulthood and return to this beach to reproduce. Darwin combined his observation with what he had seen on the voyage of the HMS Beagle, as well as with his own experiences in breeding domestic animals, and made an important association. Those individuals that possess physical, behavioral, or other attributes that help them live in the environment are more likely to survive than those that do not have these characteristics. Darwin called this process natural selection. By surviving, they gain the opportunity to pass on their favorable characteristics to their offspring. As the frequency of these characteristics will increase in the population, the nature of the population as a whole will gradually change or evolve. The driving force of evolution that Darwin identified has been referred to as survival of the fittest. However, this is not to say that the biggest or strongest always survives. These characteristics may be favorable in one environment, but less favorable in another. These organisms that are best suited to their environment survive more often and therefore produce more offspring than others in the population, and in this sense are the fittest. Darwin's theory that evolution is caused by natural selection provides a simple and direct explanation of biological diversity, or why animals are different in different places, because habitats different in their different environments and opportunities. The organisms with characteristics favored locally by natural selection will tend to vary in different places. So we'll now get to, later in the chapter, the last section, those 5 evolutionary forces that can affect biological diversity. Although natural selection is the only evolutionary force that produces adaptive changes. Darwin drafted the argument for evolution by natural selection in a preliminary manuscript in 1842. After showing the manuscript to a few of his closest scientific friends, however, Darwin put it in the jar for 16 years, turned to other research. No one knows for sure why Darwin did not publish his initial manuscript. It is very thorough and outlines his ideas in detail. The stimulus that finally brought Darwin's theory into print was an essay he received in 1858. A young English naturalist, Alfred Russel Wallace, sent the essay to Darwin from Malaysia. It concisely set forth the theory of evolution by means of natural selection, a theory Wallace had developed independently of Darwin. Like Darwin, Wallace had been greatly influenced by Malthus's 1798 book. After receiving Wallace's essay, Darwin arranged for a joint presentation of their ideas at a seminar in London. Neither attended the seminar. Wallace was still in Malaysia, and Darwin's young daughter drowned the day before the seminar took place. Darwin then expanded the 1842 manuscript he had written so long ago and submitted it for publication the following year as On the Origin of Species by Means of Natural Selection. Darwin's book appeared in November of 1859 and caused an immediate sensation. Although people had long accepted that humans closely resemble apes in many characteristics, the possibility that there might be a direct evolutionary relationship was unacceptable to many. Darwin did not actually discuss this idea in his book, but it followed directly from the principles he outlined. In a subsequent book, The Descent of Man, Darwin presented the argument directly, building a powerful case that humans and living apes have common ancestors. Many people were deeply disturbed with the suggestion that human beings were descended from the same ancestor as apes, and his book on evolution caused Darwin to become a victim of satires of his day. The cartoon in figure 14.8 is a vivid example. So Darwin's arguments for the theory of evolution by natural selection were so compelling, however, that his views were almost completely accepted among scientists after the 1860s. The evidence that Darwin presented in The Origin of Species to support his theory of evolution was strong. We'll now examine the other lines of evidence supporting Darwin's theory, including the information revealed by examining fossils, anatomical features, and molecules such as DNA and proteins. The most direct evidence for evolution is found in the fossil record. Fossils are the preserved remains, tracks, or traces of once living animals. Fossils are created when organisms become buried in sediment. The calcium in bone or other hard tissue mineralizes, and the surrounding sediment eventually hardens to form rock. Most fossils are in effect skeletons, as shown by the dinosaur fossil you'll see in figure 14.9 on page 257. In rare cases, when fossils form in very fine sediment, feathers may also be preserved. When remains are frozen or become suspended in amber, that is a fossilized plant resin, however, the entire body may be preserved. The fossils contained in layers of sedimentary rock reveal a history of life on Earth. By dating the rock in which a fossil occurs, we can get an accurate idea of how old the fossil is. Rocks are dated by measuring the amount of certain radioactive isotopes, also called radioisotopes, in the rock. A radioisotope will break down or decay into isotopes or elements. This occurs at a constant rate, and so the amount of a radioisotope present in the rock is an indication of the rock's age. If the theory of evolution is correct, then fossils we see preserved in rock should represent a history of evolutionary change. The theory makes the clear prediction that a parade of successive changes should be seen, as the first one change occurs and then another. If the theory of evolution is not correct, on the other hand, then such orderly change is not expected. To test this prediction, biologists follow a very simple procedure. Assemble a collection of fossils of a particular group of animals. So you might, for example, gather together a collection of fossils of Titanotheres, which is a hoofed animal that lived about 50 to 35 million years ago. Date each of the fossils. In dating the fossils, it is important to make no reference to what the fossil looks like. Imagine it is being concealed in a black box of rock, with only the box being dated. Order fossils by their age. Without looking at them again, place them in a series from oldest to youngest. Now finally, examine the fossils. Do the differences between fossils appear jumbled, or is there evidence of successive change as evolution predicts? Judge for yourself by using figure 1410, which traces the fossils through time from the oldest at the bottom to the more recent at the top. During the 50 million years that span by this collection of Titanothere fossils, the small bony protuberance located above the nose 50 million years ago evolved in a series of continuous changes into relatively large, blunt horns. It is important not to miss the key point of the result you see illustrated. And this again is still in figure 1410 shown now. Evolution is an observation, not a conclusion, because the dating of the samples is independent of what the samples are like. Successive change through time is a data statement, while the statement that evolution is the result of natural selection is a theory advanced by Darwin. The statement that evolution has occurred is a factual observation. Please remember that. Much of evolutionary history, vertebrates, which are animals with backbones, can be seen in the way in which their embryos develop, as I now show you figure 1411. It shows 3 different embryos early in development. As you can see, all vertebrate embryos have pharyngeal pouches or pharyngeal gill slits. All vertebrate embryos have a bony tail, or post-anal tail, even if the tail is not present fully in the fully developed animal. These relic developmental forms strongly suggest that all vertebrates share a basic set of developmental instructions. As vertebrates have evolved, the same bones are sometimes still there but put to different uses, their presence betraying their evolutionary past. For example, vertebrate forelimbs are all homologous structures. That is, although the structures and basic function of bones have diverged, they are derived from the same body part and a common ancestor. You can see this in figure 1412, how the bones of the vertebrate forelimb have been modified to make up the human forearm, wrist, and fingers, the wing and the back, the full leg of the horse, and the paddle and the fin of a porpoise. Sometimes homologous structures are put to no use at all, such as the human appendix. The great apes, our closest relatives, have an appendix that is much larger than ours, attached to their gut tube. This appendix acts as a pouch to hold bacteria used in the ape gut to digest the cellulose walls of plants it eats. Your appendix is a vestigial structure that now serves no function, as the human gut cannot digest cellulose. And again, that's a new term for you, vestigial structure. Not all similar features are homologous. Sometimes features found in different lineages come to resemble each other as a result of parallel evolutionary adaptations to similar environments. This form of evolutionary change is referred to as convergent evolution, and these similar-looking features are called analogous structures. For example, the wings of birds, or the pterosaurs and bats, are analogous structures, with different bones modified through natural selection to serve the same function, therefore produce wings that look the same. Similar function, no shared ancestry, hence those structures are analogous. Traces of our evolutionary past are also evident at the molecular level. We possess the same set of color vision genes as our ancestors, only more complex, and during early development we employ pattern formation genes that all animals share. Indeed, if you think about it, the fact that organisms have evolved from a series of simpler ancestors implies that a record of evolutionary change is present in the cells of each of us, in our DNA. According to evolutionary theory, new alleles arise from older ones by mutation and come to predominance through favorable selection. A series of evolutionary changes thus implies a continual accumulation of genetic changes in DNA. Thus, evolutionary theory makes a clear prediction. Organisms that are more distantly related should have accumulated a greater number of evolutionary differences than two species that are more closely related. This prediction is now subject to direct test. Recent DNA research allows us to directly compare the genomes of different organisms, and the result is clear. For a broad array of vertebrates, the more distantly related organisms are, the greater their genomic difference. And figure 14.3 shows this, figure 14.13 shows this well. So this is showing figure 14.14, and I'll get to that right now. So this same pattern of divergence can clearly be seen at the protein level, comparing the hemoglobin amino acid sequence to different species with the human sequence, as I'm showing you now. You can see that the species more closely related to the humans have fewer differences in the amino acid sequence of their hemoglobin. Macaques, which are primates closely related to humans, have fewer differences from humans, only 8 amino acids, than do more distantly related animals, such as dogs, which have 32 different amino acids. Non-mammalian terrestrial vertebrates differ from us even more, and marine vertebrates are the most different of all. Critics of evolution have raised a variety of objections to Darwin's theory of evolution. Darwin's theory of natural selection. So let's get to Darwin's theory of evolution by means of natural selection. So let's look at those now. Evolution is not solidly demonstrated. Evolution is just a theory. Critics point out, as if theory meant lack of knowledge, some kind of guess. Scientists, however, use the word theory in a very different sense than the general public does. Theories are solid ground of science supported with much experimental evidence, and that of which we are most certain. So few of us doubt the theory of gravity because it's just a theory. There are no fossil intermediates. No one ever saw a fin on the way to becoming a leg, critics claim, pointing to that many gaps in the fossil record in Darwin's day. Since then, however, most fossil intermediates in vertebrate evolution have indeed been found. A clear line of fossils now trace the transition between whales and hooved animals. between reptiles and mammals, and between apes and humans. The fossil evidence of evolution between major forms is compelling. And please see figure 14.15. The intelligent design argument. The organs of living creatures are too complex for a random process to have produced them. This classic argument from design was proposed nearly 200 years ago by William Paley in his book, Natural Theology. The existence of a clock is evidence of the existence of a clockmaker, Paley argues. Similarly, Darwin's critics argue that organs such as the mammalian ear is too complex to be due to blind evolution. There must have been a designer. Biologists do not agree. Complex structures like the mammalian ear evolved as a progression of slight improvements. The intermediates in the evolution of the mammalian ear are well documented in the fossil record, each favored by natural selection because each had value. Being able to amplify sound a little is better than to not be able to amplify it at all. Nor is a solution always optimal, as your own eyes attest. As you can see in the blown-up image in figure 14.16, the receptor cells in the human eye are actually facing backward to the stimulus, the light. No intelligent designer would have designed an eye backward. Evolution violates the second law of thermodynamics. For instance, a jumble of soda cans doesn't by itself jump neatly into a stack. Things become more disorganized due to random events, not more organized. Well, biologists point out that this argument ignores what the second law really states. Disorder increases in a closed system, which the Earth most certainly is not a closed system. Energy enters the biosphere from the sun, fueling life and all processes that organize it. Proteins are too improbable. Hemoglobin has 141 amino acids. The probability that the first one would be leucine is 1 in 20, and that all 141 of those, the ones that are by chance, is 1 in 20. An impossibly rare event. You cannot use probability to argue backward. The probability that a student in the classroom has a particular birthday is 1 in 365. Arguing this way, the probability that everyone in a class of 50 would have birthdays they do is 1 in 365 to the 50th power. And yet, there the class sits. So, the century-and-a-half-old intelligent design argument by William Paley has been recently articulated into a new molecular guise. Today's proponents of intelligent design now argue that the intricate molecular machinery of our cells is so elaborate that it cannot be explained by evolution from simpler stages. It is irreducibly complex. Each part of a molecular machine plays a vital role. Remove just one, they claim, and the cell molecular machine cannot function. Is blood clotting irreducibly complex? As an example of an irreducibly complex system, intelligent design advocates point out that the series of more than a dozen blood-clotting proteins that act on our body to cause blood to clot around a wound. Take out any step in that complex cascade of reactions that leads to coagulation of blood, they say, and your body's blood would leak out of a cut like water from a ruptured pipe. If dozens of different proteins all must work correctly to clot blood, how would natural selection act to fashion any one of the individual proteins? No one protein does anything on its own. No one protein does anything on its own, just as a portion of a watch doesn't tell time. Like Paley's watch, the blood clotting system must have been designed all at once as a single functioning machine. What's wrong with this argument, as evolutionary scientists have been quick to point out, is that evolution acts on the system, not its parts. Natural selection can evolve a complex system because at every stage of its evolution, the system functions. Parts of that improved function are added, and because a ladder changes, eventually become essential, in a way that the second rung of a ladder becomes essential once you've added a third. The mammalian blood clotting system, for example, has evolved in stages from much simpler systems. The core of the vertebrate clotting system, called common pathway, you'll see here highlighted in blue, evolved at the dawn of vertebrates approximately 600 million years ago and is found today in lampreys, the most primitive of fish. As vertebrates evolved, proteins were added to the clotting system, improving its efficiency. The so-called extrinsic pathway, you will see now in pink highlight, triggered by substances released from damaged tissues, was added 500 million years ago. Each step in the pathway amplifies what goes before, so adding the extrinsic pathway greatly increases the amplification and thus the sensitivity to the system. 50 million years later, a third component was added, the so-called intrinsic pathway. This is that highlighted in tan. It is triggered by the contact with jagged surfaces produced by injury. Again, amplification and sensitivity were increased to ultimately end up with blood clots formed by the cross-linking of a protein called fibrin, in which fibrinogen is the inactive form and becomes activated into fibrin. At each stage, the clotting system evolved to become more complex. Its overall performance came to depend on the added elements. Mammalian clotting, which utilizes all three pathways, no longer functions if any one of them is disabled. Blood clotting has become irreducibly complex as a result of Darwinian evolution. Intelligent design proponents claim that complex cellular and molecular processes can't be explained by Darwinism. Indeed, examination of the human genome reveals that the cluster of blood clotting genes arose through duplication of genes, with increasing amounts of change. The evolution of the blood clotting system is an observation, not a surmise. Its irreducibly complexity is a fallacy. Darwin was puzzled why dominant alleles do not drive recessive alleles out of populations. The solution to this puzzle was explained in 1908 by G.H. Hardy and W. Weinberg, who studied allele frequencies, which are the proportion of alleles of a particular type in a population. Hardy and Weinberg pointed out that in a large population, in the absence of forces that change allele frequencies, the original genotype proportions remain constant from generation to generation. Dominant alleles do not, in fact, replace recessive ones. Because their proportions do not change, the genotypes are said to be in Hardy-Weinberg equilibrium. The Hardy-Weinberg rule is viewed as a baseline to which the frequencies of alleles in the population can be compared. If allele frequencies are not changing, they are not in Hardy-Weinberg equilibrium, and the population is not evolving. If, however, allele frequencies differ greatly from what would be expected under Hardy-Weinberg equilibrium, then the population is undergoing evolutionary change. Hardy-Weinberg came to their conclusion by analyzing the frequencies of alleles in successive generations. The frequency of something is defined as the proportion of individuals with a certain characteristic compared to the entire population. Thus, the population of 1,000 cats, as you are about to see, shown in figure 14.18, and let's go to that figure now. So there's a population with 1,000 cats shown. There are 840 black cats and 160 white cats. I'd recommend you have a calculator out and prepare yourselves to practice this now, as well as do this very same on your final exam. To determine the frequency of black cats, as I teach this, what has to be done first is we have to find Q first. And you may say, but Huff, what is Q? I'll get to the equation in just a moment. So to find Q first, we will firstly find what is called Q squared. Q squared represents the frequency of the cats which are homozygous recessive and appear to be the phenotype known as white. So we divide 840 by 1,000 to get 840 divided by 1,000 there. And as that's done, the answer is 0.84. So now, the frequency of white cats is 160 divided by 1,000, which is 0.16. So the first thing we would do if we were doing this problem is to divide 160 by 1,000. That is the first thing you do. And as your textbook presented to you here on page 263, what the text gave you was the combination of all cats which were black. We cannot work out the problem correctly by knowing that ratio or that frequency. So knowing the frequency of the phenotype, one can calculate the frequency of the genotypes and alleles in the population. By convention, the frequency of the dominant of the two alleles, in this case the dominant B, is for the black allele and the letter designated by P. And the frequency of the recessive allele, the recessive B as in boy in this case, the white allele, is represented by letter Q. Because there are only two alleles, P plus Q will always equal 1. And let's get back to this in the presentation. So again, the dominant allele is always going to be represented by P, P as in Paul, and the recessive allele will always be represented by Q, Q as in Queen. So P plus Q equals 1, and that is for allele frequency. P plus Q equals 1, that is for allele frequency. So in algebraic terms, the Hardy-Weinberg equilibrium is written as an equation. For instance, we have P squared plus 2PQ plus Q squared equals 1, whereas P squared represents the frequency of those individuals which are homozygous dominant, 2PQ represents the allele frequency for those which are heterozygous, and then Q squared represents the allele frequency for those which are homozygous recessive, as I said just a bit earlier. So now let's get to agents of evolution. Many factors can alter allele frequencies, but only alter the proportions of homozygotes and heterozygotes enough to produce significant deviations from the proportions predicted by Hardy-Weinberg rule. So those 5 are as follows: mutation, migration, genetic drift, non-random mating, and selection. Let us begin. A mutation is a change in a nucleotide sequence in DNA. For example, a T nucleotide must undergo a mutation to be replaced with an A nucleotide. Mutation from one allele to another obviously can change the proportion of a particular alleles in a population, but mutation rates are generally too low to significantly alter Hardy-Weinberg proportions of common alleles. Many genes mutate 1 to 10 times per 100,000 cell divisions. Some of these mutations are harmful, whereas others are neutral, or even rarer, beneficial. Also, the mutations must affect the DNA of germ cells, the egg and sperm, or that mutation will not be passed on to offspring. The mutation rate is so slow that few populations are around long enough to accumulate significant numbers of mutations. However, no matter how rare, Mutation is the ultimate, I repeat, mutation is the ultimate source of genetic variation. Migration is defined in genetic terms as the movement of individuals between populations. It can be a powerful force, upsetting the genetic stability of natural populations. Migration includes movement of individuals into a population called immigration, or the movement of individuals out of a population called immigration. If the individuals of the newly arrived individuals differ from those already there, and if the newly arrived individuals adapt to survive to the new area and mate successfully, then the genetic composition of the receiving population may be altered. Sometimes migration is not obvious. Subtle movements include the drifting of gametes of plants or the immature stages of marine organisms from one place to another. For example, a bee can carry pollen from a flower in one population to a flower in another population. By doing this, the bee may be introducing new alleles into a population. However it occurs, migration can alter the genetic characteristics of populations and cause a population to be out of Hardy-Weinberg equilibrium. Thus, migration can cause evolutionary change. The magnitude of the effects of migration is based on two factors: the proportion of migrants in the population and the difference in the allele frequencies between migrants and the original population. The actual evolutionary impact of migration is difficult to assess and depends heavily on the selective forces prevailing at the different places where the populations occur. In small populations, the frequencies of particular alleles may be changed drastically by chance alone. In an extreme case, the individual alleles of a given gene may all be present in the few individuals and may be accidentally lost if those individuals fail to reproduce or die. This loss of individuals and their alleles is due to random events rather than the fitness of the individuals carrying those alleles. This is not to say that alleles are always lost. This is not to say that alleles are always lost, but allele frequencies may appear to change randomly, as if the frequencies were drifting. Thus, random changes in allele frequencies, known as genetic drift. A series of small populations that are isolated from one another may come to differ strongly as a result of genetic drift. When one or a few individuals migrate and become founders of a new isolated population at some distance from their origin, the alleles that they carry, even if rare in the source population, will become a significant fraction of the new population's genetic endowment. This is called the founder effect. As a result of the founder effect, rare alleles and combinations often more become common in new isolated populations. The founder effect is particularly important in evolution of organisms that occur on oceanic islands, such as the Galapagos Islands that Darwin observed. Most of the kinds of organisms that occur in such areas were probably derived from one or a few initial founders. In a similar way, isolated human populations are often dominated by genetic features that were characteristics of their founders, particularly if only a few individuals were involved initially. Individuals with certain genotypes sometimes mate with another more or less frequently than would be expected on a random basis, a phenomenon known as non-random mating. One type of non-random mating is inbreeding, or mating with relatives, such as in the self-fertilization of a flower. Inbreeding increases the proportions of individuals that are homozygous because no individuals mate with any genotype but their own. As a result, inbred populations contain more homozygous individuals than predicted by the Hardy-Weinberg rule. This is why populations of self-fertilizing plants contain so many more homozygous individuals than outcrossing plants or cross-pollinating plants, which have a higher proportion of heterozygous individuals. Non-random mating alters genotype frequencies, but not allele frequencies. The alleles are just distributed differently among the offspring. As Darwin pointed out, some individuals leave behind more progeny than others, and the likelihood that they will do so is affected by their inherited characteristics. The result of this process is called selection. and was familiar even in Darwin's day to breeders of domestic animals. In so-called artificial selection, the breeder selects for the desired characteristics. For example, mating large animals with each other to produce offspring that are larger. In natural selection, Darwin suggested that the environment plays this role, so nature decides. So with conditions in nature determining which kinds of individuals in the population are the most fit, and so affecting the proportion of genes among individuals of future populations. The environment imposes the conditions that determine the results of selection and thus the direction of evolution. So having done those, we'll next get to kinds of selection or types of selection. We'll get to three of those: stabilizing selection, disruptive selection, and directional selection. Let's get into selection. Selection operates in natural populations of a species as skill does in a football game. In any individual game, it can be difficult to predict the winner because chance can play an important role in the outcome. But over a long season, the teams with the most skillful players usually win the most games. In nature, those individuals best suited for their environments tend to win the evolutionary game by leaving the most offspring, although chance can play a major role in the life of one individual. While you cannot predict the fate of one individual, just as you cannot predict heads or tails in any coin toss, it is possible to predict which trait will tend to become more common in populations of a species. For example, it is possible to predict the proportion of heads after many coin tosses. In nature, many traits, perhaps most, are affected by more than one gene. The interactions between genes are typically complex, as you saw back in Chapter 10. For example, alleles of many different genes play a role in determining human height. In such cases, selection operates on all genes, influencing most strongly those that make the greatest contribution to the phenotype. How selection changes in a population depends on which genotypes are involved. Let's get to those three types of natural selection that have been identified, beginning with stabilizing selection. When selection acts to eliminate both extremes from an array of phenotypes, the result is an increase in the frequency of the already common intermediate phenotype. This is stabilizing selection, as shown here at the left-hand side. In effect, selection is operating to prevent change away from the middle range of values. In a classic study carried out after uncommonly severe storm of rain, sleet, and snow on February 1st, 1898, a total of 136 starving English sparrows were collected and brought to the laboratory of H.C. Bumpus at Brown University in Providence, Rhode Island. Of these, 64 died and 72 survived. Bumpus took standard measurements on all the birds. He found that among the female birds that perished were many more individuals had extreme measurements, either very large or very small. Selection had acted most strongly against these extreme sized birds. Stabilizing selection does not change which phenotype is the most common of the population. The average sized birds were already the most common phenotype, but rather makes it even more common by eliminating the extremes. Many examples similar to Bumpus's sparrows are known. In humans, infants with intermediate birth weight have the highest survival rate. Another example of stabilizing selection. Disruptive selection. In some situations, selection acts to eliminate the intermediate type, resulting in two or more extreme phenotypes becoming more common in the population. This type is called disruptive selection. A clear example is different beak sizes of the African black-bellied seedcracker finch. Populations of these birds contain individuals with large and small beaks, but very few individuals with intermediate-sized beaks. As their name implies, these birds feed on seeds, and the available seeds fall into two size categories: large and small. Only large-beaked birds, like the one on the left in the figure, can open the tough shells of large seeds, and this is figure 14.21b on page 268. Whereas birds with the smallest beaks, like the one on the right, are more adept at handling small seeds. Birds with intermediate beaks are at a disadvantage with both seed types, unable to open large seeds and too clumsy and to efficiently process small seeds. Consequently, selection acts to eliminate the intermediate phenotypes, partitioning the population into two distinct groups. There they are there, with their seeds in their beaks. When selection acts to eliminate one extreme from an array of phenotypes, the alleles determining this extreme become less frequent in the population. This form of selection is called directional selection. An experiment set up to test directional selection is shown in figure 14.21c. In the Drosophila population, flies that flew toward the light, a behavior called phototropism, were eliminated from the population. The remaining flies were mated and the experiment repeated. After 20 generations of selected mating, flies exhibited phototropism were far less frequent in the population. In the time since Darwin suggested the pivotal role of natural selection in evolution, many examples have been found in which natural selection is clearly acting to change the genetic makeup of species, just as Darwin predicted. Here we will examine two examples: sickle cell disease and industrial melanism in European moths. Sickle cell disease, which used to be called sickle cell anemia, is a hereditary disease affecting hemoglobin molecules in the blood. It was first detected in 1904 in Chicago in a blood examination of an individual complaining of tiredness. You can see the doctor's original report on page 269 in figure 14.22. The disorder arises as a result of a single nucleotide change in the gene encoding for beta hemoglobin, one of the key proteins used by red blood cells to transport oxygen. The sickle mutation changes the 6th amino acid in beta hemoglobin chain from glutamic acid to valine. The unhappy result of this change is that the nonpolar valine at position B6, protruding at the corner of the hemoglobin molecule, fits nicely into a nonpolar pocket on the opposite side of the other hemoglobin molecule. The nonpolar regions associate with each other. As the two molecule units that form still has both a B6 valine and an opposite nonpolar pocket, the other hemoglobins clump and the long chains form, as you can see in figure 14.23a. The result is a deformed, sickle-shaped red blood cell you see in figure 14.23b. In normal hemoglobin, by contrast, the polar amino acid glutamic acid occurs at B6. This polar amino acid is not attracted to the nonpolar pocket, so no hemoglobin clumping occurs, and the cell remains in the normal shape, as you see in figure 14.23c. Excuse me. Persons homozygous for the sickle cell genetic mutation in the beta hemoglobin gene often have a reduced lifespan. This is because the red blood cells that are sickled do not flow smoothly through the tiny capillaries, but instead jam up and block blood flow, which intervenes with oxygen transport. Heterozygous individuals who have both defective and normal form of the gene make enough functional hemoglobin to keep their blood cells healthy. The disorder is now known to have originated in Central Africa, where the frequency of sickle cell allele is about 0.12. So 1 in 100 people is homozygous for the defective allele and develops the fatal disorder. Sickle cell disease affects roughly 2 African Americans out of every 1,000, but is almost unknown to other racial groups. If Darwin is right and natural selection drives evolution, then why has natural selection not acted against the defective allele in Africa and eliminated it from the human population there? Why is this potential fatal allele steadily relatively common there? The answer is stabilizing selection. The defective S allele has not been eliminated from Central Africa because people who are heterozygous for sickle cell anemia are much less susceptible to malaria, one of the leading causes of death in Central Africa. Look at the maps on page 270, figure 14.24. And you will see the relationship there between sickle cell disease and malaria clearly. The upper map shows the frequency of the sickle cell allele. The darker green areas indicating a 10% to 20% frequency of the allele. The map on the bottom indicates the distribution of the malaria in the dark orange. Clearly, the areas that are colored darker green on the upper map overlap many of the darker orange areas on the bottom map. Although the population pays a higher price, the many individuals in each generation who are homozygous for sickle cell allele die. The deaths are far fewer than would occur due to malaria if the heterozygous individuals were not malaria resistant. 1 in 5 individuals, or 20%, is heterozygous and survives malaria, while 1 in 100, or 1%, is homozygous and dies of sickle cell disease. Similar inheritance patterns of sickle cell allele are found in other areas frequently exposed to malaria, such as around the Mediterranean, India, and Indonesia. Natural selection has favored sickle cell allele in Central Africa and other areas hit by malaria because the payoff in the survival of heterozygotes more than makes up for the price of death in homozygotes. This phenomenon is an example of heterozygote advantage. Stabilizing selection is thus acting on the sickle cell allele. 1. Selection tends to eliminate the sickle cell allele because of its lethal effects on homozygous individuals. And 2. Selection tends to favor the sickle cell allele because it protects heterozygotes from malaria. Like a manager balancing a store's inventory, natural selection increases the frequency of an allele as long as there is something to be gained by it, until the cost balances the benefit. Removing the heterozygote advantage. Stabilizing selection occurs because malarial resistance counterbalances lethal sickle cell disease. Malaria is a tropical disease that has essentially been eradicated in the United States since the early 1950s. And stabilizing selection has not favored sickle cell here. Well, the sickle cell allele, here. Africans brought to America several centuries ago have not gained any evolutionary advantage in all that time from being heterozygous for the sickle cell allele. There is no benefit from being resistant to malaria if there is no danger of getting malaria anyway. As a result, the selection against the sickle cell allele in America is not counterbalanced by any advantage, and the allele has become less common among African Americans than among native Africans living in Central Africa. Recent advances promise a long-time cure for sickle cell disease. To fix the defective S allele, researchers are turning to the gene editing technique CRISPR. In mice with a mutated form of the hemoglobin gene, giving them sickled red blood cells, researchers have collected immature red blood cells, corrected the damaged gene in the cells, then transplanted the CRISPR-modified cells back into the same mice. The treated mice saw no signs of sickle cell disorder. This approach using CRISPR has succeeded in correcting the S allele mutation in human blood cells in the lab. Clinical trials are underway to see if the approach works on human patients. The peppered moth, Biston nibitularia, is a European moth that rests on tree trunks during the day. Into the mid-19th century, almost every captured individual of the species had light-colored wings. From that time on, individuals with dark-colored wings increased in frequency in the moth populations near industrialized centers, until they made up almost 100% of populations of these populations. Dark individuals had a dominant allele that was present but very rare in populations until 1850. Biologists soon noticed that in industrialized regions where dark moths were common, the tree trunks were darkened almost black by the soot of pollution. Dark moths were much less conspicuous resting on them than light moths were. In addition, air pollution that was spreading in the industrialized regions had killed many of the light-colored lichens on the tree trunks, making the trunks darker. Can Darwin's theory explain the increase in the frequency of the dark allele? Why did dark moths gain survival advantage around 1850? An amateur moth collector named J.W. Tutt proposed in 1896 what became the most commonly accepted hypothesis explaining the decline of the light-colored moths. He suggested that the light forms were more visible to predators on sooty trees that had lost their lichens. Consequently, birds ate the light moths resting on trunks of darkened trees during the day. The dark forms, in contrast, were at an advantage because they were camouflaged. Although Tutt initially had no evidence, British ecologist Bernard Kettlewell tested the hypothesis in the 1950s by rearing populations of feathered moths with equal numbers of light and dark individuals. Kettlewell then released these populations in two sets of woods, one near heavily polluted Birmingham and the other in unpolluted Dorset. Kettlewell set up traps in the woods to see how many of both moths survived. To evaluate his results, he marked and then released moths with a dot of paint on the underside of their wings, but the birds could not see it. In the polluted area, Kettlewell trapped 19% of the light moths with 40% of the dark ones. This indicated that dark moths had a far better chance of surviving in these polluted woods, where the tree trunks were dark. In the relatively unpolluted Dorset, Kettlewell recovered 12.5% of the light moths, but only 6% of the dark ones. This indicated that there were tree trunks that were still light-colored. Light moths had a much better chance of survival, and Kettlewell later solidified his argument by placing dead moths on trees and filming birds looking for food. Sometimes a bird actually passed right over a moth that was the same color as its background. Industrial melanism is a term used to describe the evolutionary process in which dark individuals come to predominance over lighter ones since the Industrial Revolution as a result of natural selection. This process is widely believed to have taken place because the dark organisms are better concealed from their predators. In the habitats that have been darkened by soot and other forms of industrial pollution, as suggested by Kettlewell. Dozens of other species of moths have changed in the same way, as the peppered moth in industrialized areas throughout Eurasia and North America, with dark forms becoming more common in the mid-19th century onward as industrialization spread. As of the second half of the 20th century, with widespread implementation of pollution controls, these trends were reversing, not only for the pepper moth in many areas in England, but also for many other species of moths throughout the northern continents. These examples will provide some of the best documented instances of changes in allelic frequencies of natural populations as a result of natural selection due to specific factors in the environment. In England, the air pollution promoting industrial melanism began to reverse following the enactment of the Clean Air legislation in 1956. Beginning in 1959, the Biston population at Caldy Common outside Liverpool has been sampled each year. The frequency of the melanic or dark form dropped from a high of 94% in 1960 to a low of 90% in 1995. And you can see this shown in figure 14.26. Similar reversals have been documented at numerous other locations throughout England. The drop correlates well with the drop in air pollution, particularly with tree darkenings, sulfur dioxide, and suspended particulates. Interestingly, the same reversal of industrial melanism appears to have occurred in America during the same time that it was happening in England. Industrial melanism in the American subspecies of the pepper moth was not as widespread as England, but it has been well documented at a rural field station near Detroit. Of 576 pepper moths collected from 1959 to 1961, a total of 515 were melanic, a frequency of 89%. And then the American Clean Act passed in 1963 led to significant reductions in air pollution. So resampled in 1994, the Detroit field station pepper moth population had only 15% melanic moths. The moths in Liverpool and Detroit, both part of the same natural environment, exhibit strong evidence of natural selection. Tutt's hypothesis, widely accepted in the light of Kettlewell studies, is currently being reevaluated. The problem is that the recent selection against melanism does not appear to correlate with the changes in tree lichens. At Caldy Common, the light form of pepper moth began its increase in frequency long before the lichens began to reappear on trees. At the Detroit field station, the lichens never changed significantly as the dark moths first became dominant and then declined over the last 30 years. In fact, investigators had not been able to find pepper moths on the Detroit trees at all, whether discovered with lichens or not. So wherever the moths rest during the day, it does not appear to be on tree bark. Some evidence suggests they rest on leaves on the treetops, but no one is sure. The action of selection may depend on other differences between light and dark forms of the peppered moth, as well as their wing coloration. Researchers report, for example, a clear difference in their ability to survive as caterpillars under a variety of conditions. Perhaps natural selection is also targeting the caterpillars rather than the adults. While we can't yet say exactly what targets of selection are, the evidence is clear that selection is at work here. It is indeed one of the best documented instances of natural selection in action. A key aspect of Darwin's theory of evolution is his proposal that small-scale evolutionary changes or adaptations, called microevolution, lead ultimately to large-scale changes, leading to species formation and higher taxonomic groups, referred to as macroevolution. The way natural selection leads to the formation of new species has been thoroughly documented by biologists who have observed the stages of species-forming process, or speciation, in many different plants and animals. Speciation usually involves successive changes. First, local populations becoming increasingly specialized. Then, if they become different enough, natural selection may act to keep them away, or to keep them that way. Before we can discuss how one species gives rise to another, we need to understand what exactly a species is. The biological species concept defines a species as a group of actually or potentially interbreeding natural populations which are reproductively isolated from other groups. In other words, the biological species concept says that a species is composed of populations whose members mate with each other and produce fertile offspring, or would do so if they came in contact. Conversely, populations whose members do not mate with each other or cannot produce fertile offspring are said to be reproductively isolated and thus members of different species. What causes reproductive isolation? If organisms cannot interbreed or cannot produce fertile offspring, they clearly belong to different species. However, some populations that are considered to be separate species can interbreed and produce fertile offspring, but they ordinarily do not under natural conditions. They are still considered to be reproductively isolated in that the genes from one species generally will not be able to enter the gene pool of the other species. If you look at Table 14.2, it summarizes the steps at which barriers to successful reproduction may occur. Examine the table carefully, and we will return to it throughout the discussion on species formation. Such barriers are termed reproductive isolating mechanisms because they prevent genetic change between species. First, we'll discuss pre-zygotic isolating mechanisms and those that prevent formation of zygotes. Then we will examine post-zygotic isolating mechanisms and those that prevent the proper functioning of zygotes, the hybrid zygote, after it has formed. Even though the definition of what constitutes a species is fundamental importance of evolutionary biology, this issue has still not been completely settled and is currently the subject of considerable research and debate. For example, the biological species concept has had a number of problems. Plants of different species cross-fertilize and produce fertile hybrids at much higher frequencies than first thought. Hybridization also occurs in animal populations. This is not to say that hybridization is rampant, but it is common enough to cast doubt on whether reproductive isolation is a force maintaining the integrity of species. We'll begin there. Geographic isolation. The mechanism is perhaps the easiest to understand. Species that exist in different areas are not able to interbreed. Two populations of flowers, shown in that panel on Table 14.2, are separated by a mountain range, so would not be capable of interbreeding. If two species occur in the same area, they may utilize different proportions of the environment and thus not hybridize because they do not encounter each other. Like the lizards in the second panel of Table 14.2. One lives on the ground and the other lives in trees. Another example in nature is the ranges of lions and tigers in India. The ranges overlap until about 150 years ago. Even when they did overlap, however, there were no records of natural hybrids. Lions stayed mainly in the open grassland and hunted in groups called prides. Tigers tended to be solitary creatures of the forest. Because of ecological and behavioral differences, lions and tigers, I didn't say bears, rarely come into direct contact with each other, even though their ranges overlapped thousands of square kilometers. In Figure 14.27, it shows that hybrids are possible. The ligers, and that is a hybrid of a male lion and a female tiger. A tigun is a hybrid of a male tiger and a female lion. These matings do not occur in the wild, but can happen in an artificial environment such as a zoo. Latuca graminifolia and Latuca canadensis, two species of wild lettuce, grow together along roadsides throughout the southeastern United States. Hybrids between these two species are easily made experimentally and are completely fertile. But hybrids are rare because graminifolia flowers in early spring and canadensis flowers in summer. This is called temporal isolation and is shown in that third panel on Table 14.2. When the blooming period of these two species overlap, they do occasionally, and the two species do form hybrids, which may become locally abundant. In Chapter 21, in the next course, you will learn about that they often consider courtship and mating rituals of some animals. which tend to keep species distinct in nature, even if they inhabit the same places. This behavioral isolation is pictured on the fourth panel of Table 14.2. For example, the mallard and pintailed ducks are perhaps the two most common freshwater ducks in North America. In captivity, they produce completely fertile offspring, but in nature, they nest side by side and rarely hybridize. The branch of mating due to structural differences between related species of animals and plants is called mechanical isolation and is shown in panel 5 of Table 14.2. Flowers of related species often differ significantly in their proportions and structures. Some of these differences limit the transfer of pollen from one plant to another. For example, bees pick up pollen from one species on a certain place of their bodies, and if this area does not come in contact with the receptive structures of flowers of another plant species, the pollen is not transferred. In mammals that shed gametes directly into water, eggs and sperm are derived from different species and may not attract one another. Many land animals do not hybridize successfully because the sperm of one species may function so poorly in the reproductive tract of another that fertilization never takes place. In plants, the growth of pollen tubes may be impeded in species hybrids. In both plants and animals, the operation of isolated mechanisms prevents the union of gametes, even after mating. If hybrid mating does occur and zygotes are produced, many factors may still prevent those zygotes from developing into normal functioning fertile individuals. In hybrids, the genetic components of the two species may be so different that they cannot function together normally in embryonic development. For example, hybridization between sheep and goats usually produces embryos that die in the earliest developmental stages. If you look at figure 14.28, it shows 4 species of leopard frogs from the genus Rana and their ranges throughout North America. It was assumed for a long time that they constituted a single species. However, careful examination revealed that although the frogs appear similar, successful mating between them is rare because of problems that occur and the fertilized eggs develop. As the fertilized eggs develop, many of the hybrid combinations do not produce, even in the laboratory. Even if hybrids survive the embryo stage, however, they may not develop normally. If the hybrids are weaker than their parents, they will almost certainly be eliminated in nature. Even if they are vigorous and strong, as the case in the mule, a hybrid between the female horse and a male donkey, they may still be sterile and thus incapable of contributing to succeeding generations. Sterility may result in hybrids because of the development of sex organs may be abnormal, because chromosomes derived from the respective parents may not pair properly, or from a variety of other causes. As you continue through the chapter, please take your time to go through the Putting the Chapter to Work found on page 276. Also, I recommend you retrace the learning path through this longer chapter. And if you have any questions, please let me know.